We welcome our members and guests tonight to this evening's lecture by Greg Marshall, Vice President of Imaging at the National Geographic Society, who will be speaking on Crittercam, animal-borne imaging, a wild point of view. Greg earned a bachelor's degree in international relations from Georgetown University and a master's degree in marine science from State University of New York in Stony Brook. CNBC recently identified Greg as having one of the world's coolest jobs. And with that, please help me welcome Greg to the podium.
Thank you. Well, thank you. Can you guys hear me okay? We're going to try to, I guess, turn this off. Maybe we'll just stay away from that. Georgetown. Yeah, I like it. Thanks for coming, you guys. Yeah, it's been a few years since I was at Georgetown, but I've maintained a good connection through Janet Mann and other people that I've worked with over the years, which has been really a lot of fun. I did have a little bit of a wayward adventure up in New York, where I went to Stony Brook to do my marine science degree. It was a strange thing. I'd actually, for all my life, I've been fascinated by the ocean, and I dreamed of becoming a marine scientist, marine biologist, but I really didn't think it would be possible. One day when I was 21 years old or so, after finishing Georgetown, maybe I was 20, I just sat down and made a list of all the things that I was passionate about, and at the top of my list was marine biology. And I thought, what the heck, I'm 21 years old, why not reach out, why not try to do something, and if I fail, why not pursue this dream, and if I fail, I can do something else. But fortunately, it's what I'm going to talk about today is, in my mind at least, a wonderful life experience and a modicum of success in pursuing the dream that I had when I was a kid. And it ultimately came about with this concept of Crittercam. I was telling a couple of folks at dinner that back in 1986, when I was running a research program in Belize while I was in graduate school, I was one of the world experts on queen conch. Queen conch, it's conch ceviche. And I was studying the larval behavior and the settling behavior and the early juvenile development of queen conch. And I spent a lot of time diving in Belize. Wonderful place, incredible place to dive. But I was frustrated by the fact that very few people really cared about the research that I was doing and about the bigger conservation issue that I was trying to deal with. So I decided to make a film about the process, about the project, about the conservation issue. And it was really that effort to make a film, which I'd never made before. I had no idea how to make a film. It was really the idea of making that film that ultimately perhaps is responsible for this concept itself. I had no money. I was in graduate school, and I had to build my own underwater camera system. And it was really because I had that experience of building that underwater camera system that I was sort of positioned when the time came about to have this experience with the remora, with the shark that you mentioned, to imagine the possibility of taking this camera system that I developed and reconfiguring it, redesigning it, repatterning it, to make it look like and hopefully feel like a remora, this suckerfish, a remora that we might be able to put onto the back of that shark and ride along with the shark into its world. And so that's what I've been trying to do for, believe it or not, 25 years. It's hard to imagine. How many of you have been doing the same thing for 25 years? Well, I've been doing the same thing for 25 years. It's hard to imagine, but actually every single day is new and different and challenging and exciting. And as an example, yesterday I was in Costa Rica working with Marlin. We did Crittercam deployments for the first time on white Marlin, black Marlin on a couple of days ago. And so I actually striped Marlin yesterday. So, and it's something that we'd never been able to do before. We couldn't have done 20 years ago, 25 years ago when I first invented this or first had this idea for this concept. So I'll be talking about a wild point of view, how these animals behave, what they do out there in places that we could never hope to be personally. So we're sending remote systems into the wild. Now we've done this before. You and I, as a society, have done this before, looking out into space, and we've discovered incredible new things out beyond our little sphere of experience here on this planet. And the whole adventure of science drives us to do that, to look beyond what we know and to try to discover things that we haven't seen before, haven't experienced before, and try to come to understand how they influence our lives and how we relate to those things. But we haven't necessarily done that as well, and certainly not as extensively, in our own backyard. And we've looked way out into space with these incredible imaging systems, but what about right out there, just 50 miles, 60 miles in the Atlantic? Blue whales, humpback whales, leatherback turtles. What are these animals doing out there in that habitat, in that alien environment? It's as alien to us as outer space is. We can only spend very brief amounts of time in the habitat that these guys take for granted. But it's one of the reasons we can't be there. It's not only because we can't breathe there, but it's hard for us to see there. Half of our brain, more than half of our brain, is dedicated to processing images, to understanding our world through images. So we need to capitalize on this reality, take a look, go beyond ourselves, and see what's out there and try to experience these new things. It's an incredible planet. I mean, we were talking at dinner about the fact that now hundreds of other planets have been discovered in nearby suns. But as far as we know, this tiny little sphere of blue is the only place that is yet capable of supporting life. Probably there are millions of them out there, but to our experience, this is all we've got. And not only do we need to come to understand how this place functions and how everything is interconnected, but we've got to do it fairly soon while we're having the kinds of impacts that we're having. And that's really one of the things that continues to drive me. As it did these guys back in 1888. These 33 guys came to the Cosmos Club and decided that a new institution needed to be founded, the National Geographic Society, to explore and to disseminate knowledge to the world. And in essence, that's what I'm trying to do with this Crittercam concept, to maintain that same spirit of adventure, spirit of discovery and exploration, go out and see things, experience things, document things that we've never seen before, and then to try to share that new knowledge, those discoveries, with as many people as we possibly can. Why? Well, to get people to care. To get people to care about the fact that, yes, we are having an impact, and if we don't come to understand the impact that we're having pretty quickly, this kind of scenario is going to happen ever more often, and who knows, it's possible that polar bears won't be around 50 years from now. We need to go out and make these discoveries, make those interconnections so that we can try to prevent that from happening. And how do we do this? Well, a whole variety of mechanisms, obviously, but there's a subfield called biologging. Deploying instruments on the backs of animals, satellite transmitters, time depth recorders, GPS units, to understand how these animals move in their habitat, how they relate with one another in their various habitats, and by virtue of that, to be able to put together that really complex puzzle that sustains us all. I mean, we all are in some way dependent on the functioning of those healthy ecosystems. What do? We gather data. We gather tons of data, really incredible data. And these data are published and they're shared amongst the scientific community in droves, and it's really wonderful stuff. But there has been a problem with a lot of this data, and that is that we still have to infer, we still have to guess what these digital streams, what these digital data, these secondary or sometimes tertiary data streams really mean. For an example here, feeding events. This was a stomach temperature pill deployed in an animal, the black dots being the stomach temperature, the red line being water temperature. You see here a dip in the temperature of the stomach. The pill that's sitting down inside of this animal's stomach is telling us that something has happened there, something has changed in its environment. It's reasonable, perfectly reasonable to speculate that is the consumption of a fish, of a seal that has dove down 100 or 200 meters and has consumed a fish at that point. Might it be swallowing water? Might it be something else that we haven't imagined yet? Wouldn't it be nice to know for sure what actually happened at that event? Wouldn't it be nice if we had kind of a Rosetta Stone to be able to cross-correlate these data and to understand how to interpret those data better? And that's what the Crittercam concept does as far as I'm concerned. It gives us the ability to see things directly, use direct observation in our understanding of how these animals function. I hear and I forget. I see and I remember. I do and I understand. So how do we come to understand how these animals function? Well, this was that event that I was telling you about, 1986. This is an actual photograph. That's me. This is 25 years ago. You guys in the front row, you have no idea. We only had black and white cameras back in those days. So this was the event that occurred, that happened to me. I was out diving, had this shark swim over, a remora suckered onto the belly of the shark, came and swam around me a couple of times. We looked at each other, and the hair on the back of your neck sticks up, and you become intensely aware of your environment. But this particular event was really different. Not only had I just built this underwater camera system and sort of had that floating in the back of my mind, but this particular shark had a very large remora suckled onto its belly. And it was really in that moment that it occurred to me that if I could reconfigure that camera system, perhaps we could ride along with that shark and see how the shark's life history unfolds. Not in the 6 seconds that it's interacting with me, its behavior being affected by me being in the water, but when it leaves me, when it disappears into the mist as it did, what's it doing then? What's motivating it beyond me and beyond my experience of it for those few seconds, for 24 hours a day, 365 days a year? We needed to find a remote way of riding along with those animals and experiencing directly, observing their behavior. So here's where the 25 years comes along. 25, and some of you engineers and researchers, engineers predominantly in the audience, are going to be rather dismayed that our Crittercam systems still don't look like this. I've been trying for 25 years and we still haven't got it right. Well, two things. I mean, these, it took, you have to give me a little bit of credit. It took millions of years of evolution for these guys to get it right. The other thing is that we still haven't figured out how they have this incredible suction mechanism that they can apply to the shark's skin or a whale skin or a turtle shell and remain attached to the animal and move around. It's one of our objectives into the future, to try to come up with a mechanism for being able to not only suction down, and you'll see that we use suction cups nowadays on a variety of species, but not only to suction down, but then to be able to move around the animal's body so that you can get different perspectives. Anyway, we're still not there. One of these days. If anybody has particular knowledge of that, I'd love to talk to you later. We could work something out. So here, this beautiful thing is my very first effort to make this, to see what was possible in this world. 1987, so you'll have to forgive me. This actually is not a juvenile turtle. It's actually a fairly large turtle. And that was the first prototype of this concept. That was the best thought I could do at the time. Took a camera system, put it into a tube, tried to streamline it a little bit, and basically in a very captive environment, very controlled environment, because I didn't know if this would work or not, tested it on this turtle in the pen in Belize, and discovered, to my amazement and to the amazement of a lot of people, for 10 years after this first experiment, that amazingly this animal simply behaved, as best I could tell, as best I could measure, normally. Didn't do anything unusual. Didn't try to rub it off. Didn't try to knock it off. Didn't interact with the other animals in the pen. There are sharks and other turtles in this pen. Didn't do anything unusual. Behaved normally. And it was really that experience, really that very first experiment 25 years ago that gave me a sense of possibility, at least, that this concept might have a future. And I've been trying to make that future a reality ever since. Pretty shocking when I look back on this and think about the temerity it took to put this thing on the animal's back and to see how it would behave, see if this might work, given where we are today. But this is really what happened. The idea that you could finally give the animals the tools to show us directly what's important to them in their life history, not to take the guesswork out of it. Observation is a really key component of natural science, and to be able to actually see what's important to in the animals' lives from their perspective was and still continues to be a really compelling objective. Let me show you a quick video that is kind of a synopsis of 20, 25 years of work, I think this will work.
Finally tonight, nature as seen through the eyes of the animals. 20 years ago, a biologist wondered how sharks live when people aren't around to observe them. And out of that curiosity came the Crittercam, as it's known, a small waterproof video camera that's been used to study dozens of creatures. And right now, in Washington, scientists are sharing some amazing images. Here's ABC's Ned Potter.
An emperor penguin slides beneath the ice to look for food, and we get to go along for the ride. A penguin's eye view of life in the deep, including, watch this, a quick snack.
You're actually there with them without being obtrusive in their environment, and you're seeing things that you'd otherwise never be able to see.
Greg Marshall of National Geographic is the inventor of Crittercam, the miniature camera that allows these remarkable views from the point of view of a sperm whale, or a sea turtle, or even a grizzly bear with her family.
Lens up in the front, battery immediately behind it, and then all the electronics toward the distal end here.
It's now been used to study 60 different species. It records picture, sound, temperature, and the location and movements of the animal wearing it.
This has scientific value?
Absolutely. I mean, incredible scientific value. We're seeing things that we've never seen before.
And that knowledge may help save animals as the Hawaiian monk seal, which has been dying out for lack of food. Biologists thought it hunted just offshore, but the camera showed the seals look in much deeper waters.
Truly, as a picture's worth 1,000 words. We would never have known what they're doing until we had the camera on their backs.
A Crittercam can be attached to an animal with a harness or even a suction cup.
And if it pops loose after a few days, it floats to the surface and sends a signal for scientists to come get it.
We are now seeing a whole world of activity, animal behavior and ecology, that we would otherwise never have had the opportunity to experience.
An animal's life from the animal's point of view. Ned Potter, ABC News, Washington.
Boy, don't you want to see more of that. This is World News for this Thursday. I'm Charlie Gibson, and I hope you've had a good day. For all of us at ABC News,
good night. Well, hallelujah. Technology has continued to advance over the course of the last 25 years. That monstrosity was back the very first one. I built basically the first 4 of these in my garage or my basement or wherever. And then finally I got smart and I went down the road here to the Office of Naval Research and said, listen, these things tend to crush. They're too heavy. The guy said, listen, this was Richard Knutson down there. He was a torpedo designer. And he said, cylindrical, you'll be able to get the lightest weight, the greatest volume, and the greatest strength. So we started to design them in a cylindrical form. And right in here, in 1991, I got my first research grant from National Geographic. In 92, I got another research grant. And then in 93, as the research program began to really pick up, I became sort of full-time at National Geographic and have been at Geographic ever since. And here, somewhere in here, I was able to start hiring real engineers to do this, which is wonderful. And today, those real engineers, really a terrific engineering team at Geographic, have now made, this system was 8 inches across, this system is 2 1/4 inches across. Nowadays, we've got one of these that I just deployed on the Marlin down in Costa Rica are 1 1/4 inch and a half across. So we're getting much, much smaller all the time. Now, remember, when you think about this, and you think about the fact that you've got a camera in your iPhone, that this is a comprehensive integrated research tool. It's not just a camera. There's a lot more going on than just taking a picture. Inside this thing, yeah, there's some sort of recording mechanism. Back in the old days, we used to use videotape. Nowadays, we're using all solid state, of course. A microprocessor computer that runs the whole show, turns the camera on and off, turns the headlights on and off. And remember these red headlights later on when I show you some images of one of the species that we worked with where we got it very, very wrong, but couldn't know it until we did the first deployment, after which we realized we got it very wrong and had to change things. But we use red headlights because we think that most animals can't see red or near infrared underwater because those frequencies are absorbed really quickly. Turns out that's not true of all species. Anyway, a big battery system. Batteries today are kind of the limiting factor. solid state memory and recording capacity is growing exponentially, but batteries still are kind of lagging behind and kind of holding us back. So that's one of the challenges we face. And then ultimately, we have to recover these systems in the marine environment. In the terrestrial environment, working with bears or wolves, we can transmit off the animal in real time and experience that, what that animal's doing from a few miles away, perhaps, which we did with lions and bears. But in the marine environment, you cannot transmit this kind of data. It takes a coherent blue-green laser, and only nuclear submarines have that capacity. So we have to record all of the data in the housing, in the unit, and then recover the unit in order to get any of that data back. And we do that by having the unit released from the animal. They're slightly positively buoyant. They float back to the surface, and then you can almost see a little tip of the antenna at the very end there. That antenna sticks out of the water, sends a signal, a beacon. We have to triangulate on that and go out and pick it up. And then hopefully download the data and see something that we've not seen before, or that we can help to use as a Rosetta Stone to compare to other data that gathered in other ways. Systems today are much smaller, much more sophisticated, much more capable, much more user-friendly. One of the big pushes that we're involved with right now is to make these systems easy for researchers to use. We have potential collaborators calling us from all over the world, many times, multiple times a year. So what we want to be able to do is to give these systems to scientists to use on an ongoing basis wherever they happen to be, so that they can do their work independent of us. And by virtue of the complexity now built into these, they're very simple to use. And the idea is to get them in their hands and have them deployed over and over again. So what else are we recording in this? I said that it was an integrated instrument. It's an imaging tool, yes. Imaging, video, audio, but temperature, light levels, pressure, velocity, acceleration, magnetometry. We're gathering all kinds of data to help to contextualize that visual image and those audio data. This is an actual time depth record from an emperor penguin. Just as an example of the kinds of things that we discover. Well, when we see this, in fact, for years, my research collaborators from Scripps, Jerry Kooiman, who is the godfather of emperor penguin research, and Paul Ponganis, who is the Michael Corleone of emperor penguin research. These guys are the top of the field. For years, they've been doing time depth recorder deployments. In fact, Jerry invented the time depth recorder, and seeing profiles like this, and basically discounting all of these excursions by the emperor penguins to the undersurface of the ice, saying, oh, those are just anomalies. What's happened, all the interesting stuff must be happening down here. So when we did our first deployments with emperor penguins, we were really trying to figure out, what were they feeding on down here? And these points, these excursions to the undersurface of the ice, were essentially ignored and discounted as being not errant data, but just anomalous behavior. Well, it turns out that, in fact, that turned out to be the most interesting aspects of their foraging, their feeding behavior, their hunting behavior. Emperor penguins, fantastic animals. You've got to get to the Antarctic. You have to work with them at some point in your life. You have to experience them in some way at some point in your life. Really, really fun to work with. The easiest animal, the most difficult environment perhaps to work in, but spectacularly wonderful animals and easy to work with. So this work was done perhaps 10 years ago. Systems were much larger then. In the beginning, we used these backpack harnesses. And when I saw my son go off to school this morning, and I thought, that looks an awful lot like an emperor penguin walking off to school. But at the time, that was the best we can do. Nowadays, we can do it a bit better. Nevertheless, what we learned was really very interesting. We know that emperor penguins all dive into the water at the same time. You've seen, hopefully, National Geographic films that attest to this. Well, do they then subsequently go off and forage together? They've all dove in the water together. Do they forage and hunt together? Is it cooperative hunting? Question number one. Question number two, what is it that they're feeding on when they're diving down to depth, down to that 50 meters depth at the bottom of those time depth records? Those were the two essential questions we were trying to answer in this research program. Well, very quickly we realized they are very social around the hole, but then they are not social when they're actually hunting. And then this is what they're doing when they're hunting. They're not feeding at the bottom as we had suspected. They're going down, yes, to 50 meters deep, but they're using that as a vantage point to look up at the undersurface of the ice above them, for these silhouetted prey in the brash ice 50 meters up, 35, 40 meters up. Then they're racing up from the bottom, just like an eagle would soar above the plane and race down to get a field mouse. These guys are doing the same thing in reverse from underneath. So those excursions to the undersurface of the ice, which we had ignored as irrelevant data up to this point for the last 20 years of time-death records, that's where all the interesting behavior is happening, as it turns out. Just can't know it because you can't be there to experience it. You can't see it. It's freezing, sub-freezing water, and these guys can swim a kilometer away from in this case, this isolated dive hole. These guys were held in what's called the isolated dive hole paradigm back in those days, so they could freely swim away. Now, we did discover that there was an energetic impact, we think. They were perfectly successful. They got just as fat as the other animals, but they didn't stay underwater as long. So I was hopeful that this last expedition that we did with emperor penguins would resolve that, and it turns out that it may not have, and we still have ways to go, and I'll tell you a bit about that in a second. But how do we get these things on the animals? How do we do it appropriately? Well, you saw the emperor penguin with the backpack harness. We don't do that anymore, and I'll show you a picture in a second of what we do now. But for seals, we're pretty much constrained now to using a small patch of epoxy, and this picture's 10 years old, but it shows the epoxy, so I figured I'd show it to you. But still, we use a small patch of epoxy for seals and sea lions. For sharks, I'll show you how we do it with these dorsal fin clamps that we invented. We have to invent all these little systems because they don't exist. This is a picture taken just a month ago when I was in the Antarctic working with now, not penguins at an isolated dive hole where they have to dive through a single hole and then come back to that hole. These guys now are at the ice edge out in the wild. These are wild, free-ranging emperor penguins. This guy's just going in the water with the Crittercam system right there. And it turns out, and I don't have the images for you, I'm sorry to say, but turns out that it looks like there's still, even with this tiny little thing, I think of it as tiny, it's all relative, I guess, on its back, it still looks like there's an energetic impact that we have to be aware of and resolve in the next iteration of this thing, the next generation of this thing. But anyhow, we're perfectly successful behaviorally and in terms of their hunting behavior. And then this, I just decided I had to show you this. This was a deployment that we did just a couple of days ago on this black marlin, and with the Crittercam system there just below the dorsal fin. So, question always arises: well, how do you do this? How do you know you're not impacting the animals? Well, we spend a lot of time and energy and engineering effort as well as sort of logistical effort with our research collaborators who are world experts in their particular field to ensure that we're doing this as appropriately and with as minimal impact as possible. And it's not an easy thing to do. We understand that anytime you interact with an animal, it's likely that you're having an impact on that animal. And so we have continually sort of pushed the envelope of improving our deployment techniques. And one of those techniques goes to this issue. I don't know if you guys have worked in Shark Bay. I talked to a young lady from Georgetown who's worked in Shark Bay, Western Australia, where we did our early work with tiger sharks. And back in those days, we used to put a baited hook in the water, catch the sharks on that baited hook, bring them over to the side of the boat, flip them upside down into tonic immobility, working with Mike Heithaus, and then deploy the Crittercam systems from the side of the boat. Well, I was in South Africa working with another friend of mine, and he has spent a lot of time in the water with tiger sharks and can somehow insanely convince me to join him down there. And we got to talking about whether or not there might be a better way to deploy these systems than to catch a shark and with a hook and bring it over to the side of the boat. Might it be possible to track the sharks in close enough so that we could then reach over and use this new dorsal fin clamp that we had designed and deploy completely benignly, completely non-invasively, without ever catching the shark? So here's the Crittercam system. It's got a little kind of a clothespin type clamp mechanism. I wanted to show this because now you know that if you're ever approached by a tiger shark and it feels uncomfortable, you just put your hand on its nose. You can walk it away. All right, but make sure you have a Crittercam system in your hand, so you can then swim down and very gently, very easily pop this thing over its dorsal fin and do the deployment without ever having to handle the animal at all. Now you'll see here that this animal turns around, casually swims away, turns back in a few moments, swims back, interacts with the divers in this case, and is not disturbed. I mean, it's not unreasonable to assume or speculate that these animals are behaving naturally. Now, if you can get the divers out of the water, which you do, of course, we get the bait out of the water, which you do, of course, immediately after the deployment, then you can expect that you're seeing natural behavior. Now, there is a cost to carrying this thing. Fortunately, the systems, again, this is a few years ago, so the systems are getting smaller and smaller, and that increases our level of confidence that we are doing it, getting really valid information. Don't handicap your children by making their lives easy. So there are always animals out there that are a bit more challenging to work with, and I'm not getting in the water with a 14-foot great white shark. I'm sorry. That's just not happening yet. So we have come, it's the challenge now is, well, how do you do it? How do you do a deployment without getting in the water with an animal that, if you make one mistake, can lead to a very bad day? So we had to develop a new technique, a new system for deploying the systems. And this happens pretty quick in this video, so I'll just describe it to you real quick. You'll see a pole come out from the left, I believe it is. Out at the end of that pole is a Crittercam system that is on a mechanism that one of my engineers, Mike Shepard, developed. It's really spectacular. It has a trigger on the pole. You drop this thing very gently over the dorsal fin of this white shark as it swims past the boat. Drop it down gently, pull the trigger, it automatically cinches this dorsal fin clamp down onto the dorsal fin of the shark. And I think you'll see that the shark doesn't even seem to know that it's there. Over the dorsal fin, pull the trigger, it snaps it closed in a second. The shark just continues to munch on the boat. But it really doesn't seem to bother the animals at all. And again, clearly what's drawing the shark over to the side of the boat, of course, was the chum that you put in the water. That's why it's come over to investigate you. So you take that out, and now the shark's doing what sharks do. And as it turns out, they're not really very good at it. In all the deployments we've done, we've only ever seen them eat a couple of things, these 14, 16-foot great white sharks. And most of the time, this is what happens. They chase an animal, it disappears, and they go hungry for another day and end up eating jellyfish, which I thought was one of the most interesting discoveries that we made from working with white sharks. Well, it turns out that not only do they have a hard time feeding, but life history of a shark is pretty tough, and the broader context of why we do this is always important. I mean, we don't just do this. Well, yes, we do it for the exploration, for the discovery, for the science, but there's also a really compelling conservation narrative. I mean, you and I, each of us, every single day has tremendous impact on this planet, and we really need to begin to understand what that impact is and to begin to moderate it wherever we can. Truth is, we are, in many respects, we sort of lose ourselves from time to time. But we are the genius of our species is that we care. We care whether or not, these little tiny turtles exist or not, and whether or not they have a future. We care, and that's really what's wonderful about our species. We're not so stupid after all. And by virtue of that caring, we're willing to invest in the hard work to find out if we can make the kinds of discoveries that we can do. This is Richard Reyna, one of the world experts on leatherback turtles we worked with for years. And so we did a series of deployments testing out this new suction cup system that we developed to see if it would work. And so I asked Rich, I said, okay, Rich, well, leatherback turtles can dive down 1,000 meters. Just, here's the Crittercam system. We'll pop it on your back. Now just get down to 1,000 meters and report back how it feels. He said it felt fine after 10 meters. Anyway, so we always, wherever we can, and we can't always do this, wherever we can, we work with animals in a controlled environment first. These are the beluga whales up at Mystic Aquarium up in Connecticut. And we have a project with belugas. We were hoping to start it last May. It'll start now this May. But we did a variety of tests to see what's reasonable. How will this work? Is it reasonable to deploy it? What we'd really love to see, of course, is what's happening in the mouth. We'd love to be able to deploy it right here and look across the eye and across the mouth to see exactly what's going in most easily. Well, turns out when you deploy it over here, when you deploy that suction cup over here, they tend to swim in a circle. And they tend to look back at it. So it's obvious that that's not going to work in the wild with the wild animal. We discovered that with a captive animal. So, but up here on the back of the back, they just don't seem to care. And while it may not be the ideal perspective, it's the best that we can get with an animal in order to minimize the possibility, the likelihood that in some way we're disturbing or changing its behavior. Although in this case we couldn't really see that there was any impact at all. All truths are easy to understand once they're discovered. The point is to discover them, and that takes a little bit more work. And the work, of course, happens not in the lab, not in those controlled environments, but out in the wild. And so that's ultimately where all of our work takes us, is out into these field situations, working with animals that our research collaborators, and these are all research collaborations, as I mentioned, have done a lot of work with, but have really not, but still have lots of questions for. So, for example, these hawksbill turtles in Mona Island in Puerto Rico, they're looking at the mating behavior. This is a few years ago now, but it was a simple question. How do the males find the females so that they can successfully mate and reproduce? Well, it turns out, in all the Crittercam deployments that we did, and we work with the males because they're sort of the expendable component of the population, as you guys know, these male turtles were fantastic at finding other male turtles. Never once did they find a female turtle. It's like, what is going on in this system? How can this system possibly work? So we decided eventually, after a bunch of deployments, that we had to deploy at least one on a female turtle to see what she was doing. And this is what a female turtle does all day. She hides in a cave. 3 days long, diving down, sitting in a cave, avoiding the males, and then just surfacing to breathe and back down into the cave. So how that mating system works, we have no idea. We thought, well, I'm generous when I say we. I mean, the guys, Richard Reyna, Jim Spatilla, Frank Palladino, the world experts on leatherback turtles, thought we had a pretty good handle on what their mating behavior was like. Females store sperm, they probably mate out at sea. All the genetics suggested it was one male, probably one encounter. And then the female swims on shore, fertilizes the eggs at will, lays the eggs, and then goes back to sea. The males, 1,000 miles away perhaps, out in the pelagic zone, never have to go through that traumatic excursion to the fishery zone. So in this case, we didn't think the mating behavior was interesting. We thought that their foraging behavior would be an interesting question here. What are they feeding on to help sustain these females, to help sustain them through this really energetically demanding period of nesting, when they're laying eggs, clutch after clutch after clutch of eggs? We never expected to see this. Right offshore, right off the nesting beaches, that long tail indicates that that's a male turtle, male leatherback turtle. The male turtles, as it turns out, have made that same migration from the pelagic zone, following the females into the nesting beach, and they're engaged in what we think is mating behavior. Now, once again, in this case, the Crittercam system may have interrupted the success of those mating encounters. We didn't expect the animals to, the males to be there. But actually, another interesting aside is the females don't seem to be very interested in these males. But the fact that we've now demonstrated, we've now proven that what we had thought for years about their mating behavior happening maybe 1,000 miles in the pelagic zone is just simply not true. The males are there, they're engaged in mating behavior, and we need to rethink, not only that aspect of their ecology, but also the management now, because the males need to be part of the management equation all of a sudden, which we didn't have to worry about before. Green turtles, all over the world, they're supposed to go through a transition period from being omnivores, eating everything, plants and animals, to being herbivores, 3 years of age. Well, they spend most of their time, adult green turtles spend most of their time eating jellyfish. Not a plant, it's an animal. And we just don't, we now know this, but when we first discovered this 10 years ago, we understood all of a sudden that, it's because you don't see it when an animal washes up on the beach. The jellyfish is just disintegrated into water, or you do lavage, it's just turned into water. So we're just discovering all these fundamental things that we just can't know unless you're there to observe it directly. And in some cases, we do all the statistics, and visual information is very rich. You know exactly how the animals are diving. You know the exact durations. You know how many breaths they're taking at the surface. So you can parse all of these individual data parameters out, and make new discoveries about new types of. Are any of you in the sea turtle community? Well, good. I can make stuff up then. No, but I mean, there's huge discussion about what the dive profiles, dive patterns mean. And I mean, first of all, these 5 patterns were well documented by time depth records. And so really the question was, what are they doing in each of these dive types? Well, nowadays, not only did we discover a whole new category of dive type, but also, we know pretty much exactly what they're doing. There's no, it kind of takes the fun out of the science, really, because you can't speculate anymore. This is what they're doing. But that's kind of the great thing about science, is we're trying to get to the point where we actually know something. Anyway, exploration really is the essence of the human spirit. To get out, look beyond ourselves, to explore that new thing, and to try to work with the most homely of animals. It's really our responsibility to do this. Just kidding. Seals and sea lions, wonderful animals. Harbor seals, this work quite a few years ago now. Really clumsy on land. But once they get in the water, just these spectacular balletic creatures. In this case, we really were looking at the mating behavior. These guys were suspected of having set up leks, the first underwater animal to actually set up lek mating system. Well, it turns out you can't not learn almost everything about their behavior, including their foraging behavior. Now, you saw first feeding on schooling fish. Other animals idiosyncratically feed on bottom fish. Well, when you're sucking a fish out of the sandy bottom, you're going to get a stomach full of sand. What do you do about it? Well, you go up into the water column, you regurgitate, and then at your leisure, after the sand is filtered out, you go back and you feed for real. Now, this one piece of the puzzle is not going to change the world, but it's a fascinating new insight into an aspect of behavior and ecology we'd never imagined before. Hawaiian monk seals, probably the best example for us of a species we've worked with that's critically endangered, and that the kind of information that we could gather would really help, specifically in the management of the animals. We had thought for these guys, and again, generic we, that in fact 20, 30 years ago, we had thought that these atolls that they live were probably not only their critical social habitats, but also their critical foraging habitats. So protect the atolls, the shallow waters of the atolls, which are rich with fish, and you can protect the species and give them a chance into the future. So for years, the shallow waters of the atolls have been protected since the early 70s. The problem is that the numbers continue to decline. And we hadn't known why, and I'm not saying we absolutely know why today, but when we did these Crittercam deployments, we discovered very quickly what's going on here. Yes, the seals are socially active in these shallow waters of the atolls, but they never feed there. When they go feeding, when they go hunting, when they get serious about sustaining themselves, they went outside those protected boundaries into these deep, what appear to us to be relatively barren waters, and that's really where they do most of their behavior. Now, this is just an example of what we're always looking for to help give us confidence that we're not affecting the animals in a deleterious way. That other animal didn't have a Crittercam system on his back and was rubbing on the bottom just like the animal with it. Anyway, what happens is this is down in this habitat 300 feet deep, 400 feet deep. They're down there flipping over these rocks and evicting the fish that are octopus that are hiding underneath these rocks. And it's in that habitat, those unprotected waters, that they're doing almost all of their feeding behavior. In the process, they're creating a foraging community, competitive, kleptoparasitic community of not only themselves, they're creating the opportunities, but sharks are coming in and the big jacks are coming in and trying to steal their prey. So what they do is they grab the fish and they eat it on the bottom if they can, but if they have to, they go back to the surface and eat it at their leisure there when the sharks and the jacks stay on the bottom. But we can know exactly how much time they're spending feeding on exactly what species. So we're kind of taking the guesswork out of it, which is really great. And then, of course, again, being able to parse out exactly how much time, at what depths they're creating these other foraging opportunities with other species. Whales, I'm sure you guys have all seen whales. They disappear and it's like, wow, magnificent creature. What are they doing down there? How are they functioning? How are they surviving? How are they communicating with each other? Well, in the beginning, I probably chose the wrong animal to work with, which was the sperm whales, but really magnificent animals. And back in those days, we just didn't have a better way to do it than to kayak up to the whales, these sperm whales, and lean over the side of the kayak and deploy the Crittercam systems on their back with a suction cup. Really wonderful animals. At this point, this is quite a few years ago now. We had really no idea what the response of the animals would be. But anyhow, let me let you listen to this, if this plays. Very social, very interactive, very tactile with one another. Down it. This is relatively shallow, but it's the first time that anyone had ever known that a calf, this is a calf, you know that now because it doesn't have the dentation fully developed, down at those depths. And then deeper yet still, another animal coming in to investigate and to socialize with this animal, with the Crittercam system on its back. Someday, we'll know what those clicks mean. Someday, because we can put together the image with the audio, correlate the two, behavior response, someday we'll be able to parse out, maybe it's you guys, be able to parse out what that means, the language we were talking about at dinner. We can't do it now, but someday. So, humpback whales, Southeast Alaska. Magnificent experience, but you can't kayak up to these guys. So again, we had to develop a new way of doing it. We had to develop a suction cup system. We could lean over the side of the boat, automatically deploy these things on the backs of whales. And working with Fred Sharp, who's one of the world experts on this phenomenon called bubble net feeding, which is really, really spectacular. Group of animals gets together, 12, 15, 18 animals, humpback whales, and they form this collaboration. It's almost a ballet. It's almost like Fred describes it as a football team of whales with very distinct individual jobs to do to corral these herring in a ball at the surface so they can engulf them as they race up to the surface. Now, he's been studying this for 15 years, but had never been able to see it. He's been studying it acoustically and had a pretty good idea of what was going on, but didn't know, for example, that they go to the bottom to flush the fish off the bottom. We now know that because we can see it. Didn't know whether or not they used their pectoral fins in the process. And it now looks like they do. That white undersurface of the humpback whale's pectoral fins, everyone's always wondered, well, why do they have that? Well, it looks like they may be using that to flash the fish sort of into their mouths. Scare the fish away from that white pectoral fin into your mouth. I don't know if I have audio here, but trying to scare the fish into a boat. Well, the interesting thing is that also one of the things that we discovered, which was sort of a negative result, is that they're actually pretty lousy at hunting. A lot of the fish escape, and that was one of the questions was, well, how is it if we imagine that they're getting 500 pounds of fish in every lunge that they do, and they lunge 30 times a day for 2 months at a time, how can they possibly be eating that much? Well, it turns out a lot of the fish are able to escape, and they're not as effective at it, not as successful as we'd imagine. So, again, it's a kind of a negative result, but again, it helps to explain that the breadth of their ecology. Writing down to the bottom, we've got projects now in the Antarctic as well. Killer whales, we've only done a couple of deployments on killer whales, and I think it's one of the things that's really exciting, complex, highly social animal. I really didn't think this would work at all because of that. I thought they would communicate with one another. The strange thing on my back, please get it off. But they didn't seem to care at all, which was really surprising. Didn't seem to care that it was there. And so it's politically complex to work with these guys, so we have yet to do it. But one day we will, and it's going to be just incredibly exciting, the kinds of things that we can learn. We haven't worked with any, except for the killer whales, we haven't worked with any of the dolphin species, the smaller species, in part because our systems haven't been small enough yet. And today I think we're at a point where they are small enough where we can reasonably expect to not only not change their behavior, not disturb their behavior, but get interesting, valuable information. So I'm trying to convince these guys who are working in Shark Bay in Australia with Janet Mann to maybe think about doing it. So go take that message back to Janet. Humpback whales, I think I showed you. Okay, so we've got these suction cup systems. And I just wanted to show you briefly some of the work we've done with these guys in Maui. Whole different scenario. The first clip I showed you was in Southeast Alaska. Now they're in Maui, where they're engaged in this mating behavior. And we think of humpbacks. How many people think of humpbacks as being these genteel, wonderful, charming animals? Come on now, be honest. There we go. Good. Well, there's a dark side to humpback whales too, and it happens when they're mating. And the males are seriously aggressive and rowdy. And that's what they're doing here. They're banging into each other underwater where we can't see it. They're extremely aggressive, beating each other up so that they can gain access to that as that primary escort for the female to create, hopefully, the opportunity then to mate with her. It's really about showing prowess. And we seem to be seeing, and again, this hasn't yet been confirmed yet, but we seem to be seeing that they're also using bubble blowing as part of that display. And the speculation is that if they can blow bubbles while they're engaged in this really aggressive behavior, well, that sort of demonstrates excess capacity. You know, you're not afraid of sinking. You're not afraid of having to go to the surface. You're big enough to be able to waste some air underwater. So we're seeing that, but we're also seeing bubble blowing in a really also another interesting aspect of the behavior. Here, once the animal has succeeded, it's gained that primary position with the female. This now is the female. Here's that primary escort. Bubble blowing in a new way now. And he comes underneath here. All's very gentle now. He comes underneath and continues to blow this nice, gentle stream of bubbles right underneath her until, of course, some interloper comes along off screen and he races off for the moment. You see this here for the moment. He races off to chase away this other male that's come in to try to interrupt his courtship, scares that other animal off, then goes back to the female who's still hanging in the water there and does something really, really cool. Now, we don't know exactly what this is, but I think it's interesting. Now watch what's happening here. She's hanging in the water. He goes underneath her and blows a stream of bubbles. Those bubbles hit her on the belly, rise up, and just caress her as she's hanging in the water. Now, is that a precursor to, the next step? I have to imagine we haven't seen that, but this is an important part of their behavior and not seen before. Ultimately, we think that culminates in something like this. We've done one deployment on what turned out to be a calf, small whale, 20 feet long, not 40 feet long. And we were able to demonstrate conclusively that it was a calf with what you're about to see. Never documented before, never, I don't really think that anyone's ever seen this before. Nursing, sloppy feeding, as much milk going, into the water is probably that calf is getting quite a bit of milk being forced into its mouth. But so mom and calf. Now, what happens here, and I can't do it justice because it's brief, and I know we don't have time, but watch what happens. The mom taps the calf on the head, and basically my interpretation of that is, okay, Johnny, I got to go feed now. You had your dinner. It's my turn. She goes down. The calf follow me down, and again, yes, anthropomorphic, but play with me here. This is now the calf watching this bubble net feeding event unfolds from underneath, learning, perhaps, how to do this. All these animals racing to the surface, screaming. Look at all this herring that are escaping. You could tell that they're lousy feeders. But, is there learning happening there? Sure looks like it. Sure looks like it. That's the great thing about this. You get these tantalizing insights, and you need to do more work. We're all part of this equation when we've got to go. We've got to take all these animals seriously, including this guy. And I remember that red headlights. Okay, you'll see that we don't always get it right, and in this case, we got it very wrong. Humboldt squid, Sea Cortez, red headlights, first deployment, only the first deployment. Evening deployment, it finally goes down into the darkness, and the red headlights come on. And guess what happens? Well, You'll see in a second. Bad things happen because of those red headlights, and we know this because of subsequent deployments that we did. It was absolutely relentlessly attacked by other squid down there. Until finally, and this has only happened, in a course of 4 or 5, 6 minutes, they finally were able to pull the camera, the Crittercam system, off the squid. It then started to float back to the surface, and they attacked it relentlessly all the way to the surface. And you'll see here in a second, there it is, it's floating free now. This guy's got it, and you're in the mouth of a Humboldt squid. So bring that home and have a pleasant night's sleep. And now, how do we know that this was the consequence of the headlights? Well, we knew something was awry here. When we got the first deployment back, we thought, well, this something must be strange here. Maybe it's the headlights. So the next deployment was this. This was daytime deployment now. No headlights, ambient light welling down from the surface. Completely different behavior, completely different response, and I think it's pretty reasonable to conclude that it was the headlights themselves that caused that very aggressive response on the part of the other squid. Now the cool thing here is we don't know exactly what's going on, but all of this flashing is not downwelling light from the surface. This is communication between these squid, and again, we have no idea what it means, but someday, because we can correlate now what we're seeing with the response of other animals, it gives us the hope that one of these days we'll be able to understand exactly what that means. They're beautiful. I mean, who knew that they use their tentacles as ailerons, as wings down there? We just had no idea. And that should go. All right, last one, I think. How are we doing for time? Am I over time? Are we okay? Okay, well, let me just get through this real quick. Alligators, we've done a little bit of work with alligators down in Florida. Actually, it was one of the first animals that I ever worked with. And I just wanted to show you one really kind of fun little thing that I don't have an explanation for yet, but it is very intriguing. It's not something we would have expected. So, the alligator sits around, and it becomes a good feeding station for other cleaner fish. But then this next scene is really weird. What happens is the alligators, I have to describe it for you a little bit. It's sitting at the surface, breathing at the surface, and then all of a sudden drops from the surface down to the bottom. And you see it's dropping right now, bang, hits the bottom, is upside down, it bounces off the bottom. You can see it's not really moving, it's just bounced off by inertia, it's bounced off the bottom, lands on its other side, bang, upside down. Now we edited this because it stayed in that position for 45 minutes, upside down on the bottom. Like, what is going on? So we've edited this just so that we don't have to sit you here for 45 minutes. All of a sudden wakes up, realizes that it's going anoxic or hypoxic, races to the surface to get a breath. Now, well, yeah, who knows? But who would have ever expected that to be sort of in the behavioral repertoire or physiological repertoire of an alligator? We don't have any idea what's going on there, but it's intriguing, and that's the kind of discovery that makes this this 25 years so exciting all the time. Now, a magnificent butterfly finds a little heap of dirt and sits still on it, but man will never on his heap of mud keep still. We're always pushing. We're always intrigued by that next challenge. And thank God we've got our little house cats and house pets to help us with this process. This is Snake. She's my cat. She's the first volunteer to carry a terrestrial version of this system. In this case, we could now transmit the images off the animal directly and view what's going on. And so Snake was not, she's a real homebody, so she was great as a test animal and did fine. But Molly was something else entirely, and I figured you guys might be interested in seeing Molly. Molly's a lovely cat. How many of you guys are cat owners? Yeah. Love your cats, snuggle with your cats, kiss your cats? I did. Molly was the same way, and her owners did all those things and loved her. But she lived in the back streets of Washington, D.C., and they knew that she went out into the streets of D.C. at night from time to time and brought home gifts. So, we figured that we'd see how she went out and secured these gifts for her owners. So we deployed the system on Molly. Now we're 2 blocks away seeing this in real time. Incredible animals, incredible predators, of course. And really, even though she's a nice little genteel domestic cat, really good at what she does, and you'll see that in a second. Well-fed animals, got a big bowl of food at home, and still she does this. Of course, she's just going to catch it and bring it up to the stoop. You can see that she's got the rat. Brings it up to the stoop, and okay, all right, but you can kind of understand she's going to leave it there on the stoop, and in the morning we're going to wake up and get it. And thanks a lot, Molly. We appreciate that. But then the system continues to transmit and continues to roll, and you start thinking, what is going on here? She's not doing what we're not seeing this, are we? She has a full bowl of cat food sitting just across the door. Somewhere in here, I'm thinking, maybe I'm not going to kiss my cat anymore. And right about here, I'm dead sure of it. All the way down the hatch. I don't know. There are things we don't know about animal behavior. So this is what the system looks like today. It's a very small system. This is my new cat. She's the next volunteer. And we've got a long ongoing project with the University of Georgia to deploy these on lots and lots of house cats. We've got thousands of hours of images now that are being analyzed by the graduate students down there to see what impact our house cats have on songbird populations out in the backyard. So it's a really important study. Somewhere, something incredible is waiting to be known. We've worked a bunch of different places around the world, but what's really intriguing to me, is all the stuff, all the incredible things that are happening all over the planet that we don't know anything, that have yet to be discovered. Every single deployment that we do shows us something we never imagined. Not every deployment, but many deployments shows things we've never imagined, and all the incredible stuff yet to be discovered out there. All these different species that we just don't know we don't know much about, and we haven't been able to directly observe them in their natural environment. So we've got a long, long way to go. As the technology continues to get better, continues to get smaller, it just creates more and more opportunity to work with ever smaller species. We've been able to work with the big guys now, but that's just the bottom of the curve. What about all those other animals that we just haven't been able to? That's a great face. We haven't been able to work with. So there's a long, long way to go with this, and despite all the distance that we've come so far, okay, enough of this. All right, let's get going. And it really will. It comes down to the new technologies. As technology continues to progress, it gives us the chance to work with animals like this Colugo flying squirrel. Now, first thing I think I mentioned that we do controlled experiments first. Work with animals in captivity wherever we can, and that's what this is. This is a technical experiment. This little tiny new camera system that my guys at Geographic in the lab developed. Well, first of all, is it okay? Is it appropriate? Can the animal function? Can it climb? And then, when it finally gets down to what it's known for, this leaping and flying behavior, will it work? Will it be functional? And, seeing what you're about to see gives me, kind of the hope that, one of these days, I just have to show this again, it's just too much fun, so watch this in slow motion now. Someday not too distant future, we'll be able to work with these guys in the wild, and we'll be able to see what's going on up in the canopy where we can't see them. So exciting new possibilities and opportunities as the technology continues to advance. Well, all this stuff is published. We've published, 35, 40 papers at this point. And it's really important to share this information with peers and to be reviewed by the community. So publication is an extremely important part of what we do. But it's not enough. And the end of life is not knowledge, but action. What do you do with it? How do you sort of have an impact and change the world and get to those bigger conservation issues that we're talking about? And so every project that we do, we're filming everything that we do with the hope that someday these projects turn into something that we can share with hopefully millions of people and get people to do what National Geographic does, try to inspire people to care about the planet. And we've made 70 films of various types, and continue to do so. We also try to get together and get directly down and connect with a variety of different communities and try to teach kids how to do these things so they can get inspired by science and conservation as well, directly by being involved with the work directly, getting their hands dirty. And then using all the vehicles at National Geographic to get out and to share these stories, share the excitement of the science and the research with the community at large. In the end, we'll conserve what we love, and we'll only love what we understand. And the concept behind Crittercam is to help us get into the spaces on this planet that we otherwise can't get to, and understand things that we otherwise couldn't understand, and see it directly, resolve some of those conundrums that we've been fighting with in our own minds and in our research programs for years, and basically to give the animals themselves the tools to tell us what's important in their life histories. Thank you. Thank you. Thank you very much. Yes?
Yeah. I was intrigued about this whole presentation. It's very nice. I enjoyed it a lot. I saw a presentation on PBS this last week on critter camps on kangaroos.
Mm-hmm.
Very informative as far as what the kangaroos were actually doing. The first part of my question is, how many critter camps are now out there, and what do they cost? The second part is, I'm interested in creating a position at a nonprofit called the Marine Hunting Institute. We need to find more places where we can have impact on them.
Fantastic, fantastic. Today, my dream, and I think I mentioned this briefly, is that we can get these in the hands of any scientist who has a question that they feel that these systems can help answer. Our challenge is everybody's challenge. There's never enough money. There's never enough time to do all that you want. So today we have somewhere on the order of, a mere 10 or 15 Crittercam systems that are, probably 20 Crittercam systems that are being deployed in a variety of projects right now. It should be 100. In my mind, it should be 500. And there's just a little disconnect between the objective and the financial reality. But they're not that expensive these days. Back in the old days, when I first developed this, of course, we did a huge amount of engineering time to make a single system work. And then it would take a huge effort on the part of someone well-trained, myself, my research partners, to make these systems successfully deployed in the field, to enable them to be successfully deployed. Today, because our engineers are so much more capable, the systems are much more sophisticated but simpler to use. And so they're also can be built much more cost effectively. So today we can build a system for $5,000. If we could get to that 500 unit mass production, we'd get them down to $1,000. And we'll get there. It's just a matter of time. We have a number of people have now tried. It's actually turns out to be fairly difficult. So there are a couple of people now who have had some success.
You didn't hear the question.
Oh, I'm sorry. I should repeat the question. Yeah, are there competitors? And the answer is yes, increasingly so, I think. Over the years, there have been just a few people who have tried to do this, and one of them being Randy Davis and his team at the University of Texas. And he's done some great work with Weddell seals down in the Antarctic. There haven't been many other people. However, today, because camera systems are getting so small, there are people who are taking small cameras, like you probably know about the GoPro camera, the GoPro Hero camera. It's a very small, incredible camera. Everyone needs to go out tomorrow and buy one because you can't afford not to have one. $300 for a tiny, beautiful, very elegant, high-definition camera system. They're literally twice the size of this. Well, you can take that, and people are now, in my mind, somewhat unfortunately, taking just the camera and strapping it onto an animal's collar or something like that for the purpose of making television. And I think it's not what we do, but it's becoming more feasible because of the technology. Yeah, next question. What was it on the whale?
What were the circles on the whale?
Oh, what were the circles on the whale? Great question. Well, you know, if it's a sperm whale, you probably know what those circles are. When the sperm whale dives down to 1,000 meters deep, actually probably only 400 meters deep, we now think, there are incredible squid down there called giant squid, Architeuthis, that are 60 feet long, almost the length of this room maybe, with their tentacles. And the thought is that they have these titanic battles with the sperm whales that are trying to eat them. And so the squid, we think, we've never seen this, no one's ever documented this. We tried, but we didn't succeed entirely. We think that the squid are wrapping their tentacles around the body of the whale, trying to keep from going into the whale's mouth. So we think that's what those circles are on the whale's body. Yeah?
Yes, that's true. Go ahead. Yes. No, it's sorry. That would be next.
Thank you very much. With each development in astrophysics, you know, the more Mars explore, everything gets a little more sophisticated, a little bit more. What would you like to see Crittercam doesn't have now? But if you had unlimited funds, what would you have on Crittercam?
So the question is, if we had unlimited funds, what would we like to see in the Crittercam systems now? You know, where would we like to take it? There are two parts of that question. The first part is that there are 50 research programs, maybe more, you know, probably 100 research programs around the world that would like the systems as they are today to get them in the field. And my first objective would be to satisfy that immediate need. Now, once that were satisfied, the next thing is really comes down to data gathering capacity. And there's a constant struggle. You know, we're a research program, but National Geographic's also very much a communications media company with the intent of inspiring people to care. And so having a high definition capability in these systems becomes increasingly important. Of course, there's a trade-off, because the more high definition, the higher quality your image is, the less recording capacity you've got. So the scientists are saying, you know, we want 20 hours of recording capacity. High definition gives us 10. So I guess the thing would be to increase the recording capacity, high definition, but that requires a better battery, a new kind of battery. Lithium polymer, lithium-ion batteries that we use right now are incredible batteries, but they're still not quite enough. And then, of course, the additional thing is that the images and the audio that we gather have, in my mind and in the research community mind, have to be integrated into an environmental context. So we do have magnetometers on, we do have accelerometers on. We need to take the next step of fully integrating all of these parameters into a more user-friendly, more tractable, more manageable visualization package, so that you can put it all together in real time.
For the smaller marine animals, you appear to increase their drag considerably, and I'm wondering if you had a guideline for the maximum increase in the drag coefficient, that percent increase that you were willing to tolerate. I think 10%.
Good question. The question is, for the smaller animals we work with, we seem to have an energetic impact on them by virtue of the drag component. And is there a guideline that we have that defines which animals are appropriate to work with and which are not? The truth is that we don't, and that there isn't one that we know of, that I know of today. For me, it's a gut feeling, and that's not much to go on. But for example, we do tests with dolphins. Previous incarnation of the Crittercam system, we did tests with captive animals with dolphins, and it didn't feel right, didn't look right, it looked like there was some impact. That it was almost just a gestalt rather than a quantifiable number. So no, we don't have a great guideline. We do have a guideline in terms of the weight of the system, but unless, maybe you guys know, maybe there's something that is out today that I'm not aware of in terms of a drag coefficient guideline for marine animals. It's a big concern. There are people who are doing this now, who are measuring the drag coefficients of various instruments that are deployed. I guess all I can say is that we are very, very aggressive to minimize the impact and to only work with animals that don't seem to change their behavior once we've done a deployment. And we're looking at that very aggressively. So there are a number of species, dolphins being one example, but we haven't continued to work with because just it didn't look right. Yeah.
Okay, you mentioned tangentially that the seals you observed might have a left mating system.
Mm-hmm.
Were you able to observe this, and so what was their behavior like?
Nope, we didn't. It didn't. Yep, it's still speculative. We didn't happen to see that over the course of the many deployments that we did. We did 45, I think ultimately about 70 deployments on those harbor seals, and there wasn't enough information to be able to demonstrate that. So we still don't know. Has yet to be done. Yeah.
There are some similar technologies being used on more willing subjects, like in particular military applications or police applications, or just a creative individual who carries along a video camera for 24 hours, documenting their life. How does those kinds of activities intersect with what you're doing? And more specifically, you seem to be taking views from one camera lens. How important is gathering data that gathers sort of spatial relationships, either a very good stereo or maybe something like the hydro effect of having ways of understanding how animals are perceiving spatial things going around?
Yeah, two great questions. First is how are we interfacing with potential military and or rescue applications of these kinds of technologies, imaging technologies, and then two, are we gathering or do we aspire to gather more spatial information about how these animals are positioning themselves in the environment? And yes, to answer the first question is we have tried. I mean, very early on, I realized that we need to deploy these systems on rescue dogs, for example. The problem is, and we haven't done it yet. It's hard to believe. We actually talked to the guys at Fairfax who travel around the world after earthquakes and deploy to affect those rescues. And it makes perfect sense that the dogs that they carry out there should carry cameras. Technology really hasn't been there until very recently in terms of the size of package that would be appropriate to deploy. So it makes perfect sense to do it. We haven't yet done it. Other people are going to catch up though, and I've just talked to a guy over the weekend. In fact, he's the person who sponsored most of the research on the billfish down in Costa Rica. He has a military and a police background, and he said that his officers are now starting to carry cameras on their shoulders, every single one, to protect them from accusations of impropriety. And so it's going to be a more and more important part of law enforcement as we go into the future. Unfortunately, we're not participating in that at this point, although we should. The other thing is spatial information. Yeah, we would love to do stereo. We would love to gather. We are gathering that three-dimensional trajectory information. But that doesn't provide what I think you're talking about in terms of the image, even if it's acoustic, some sort of way to image the environment in a three-dimensional way. And we haven't got that. It's simply a function of size and power capacity and power limitations that we face with these systems. We're constantly trying to make these systems smaller so that we can more appropriately work with the animals we're already working with. And to double it has been a difficult trajectory to go on up to this point. Love the idea, we just haven't quite gotten there yet in terms of reducing the size of these enough.
Any other questions? Yes.
Well, just a comment. I've got many gigabytes of GoPro video. It's fantastic. The question is, do you have a high loss rate when you attach these cameras and they release after time? Do you lose like 10 or 20% or just 5%?
What's our loss rate on these systems? And it's something I've camouflaged. I wanted to keep from you guys, so thank you very much for asking that question. Anyone here from National Geographic? Anyone? No, and remarkably, it's actually not been that bad. When I first started this, I thought for sure we'd lose every other one. There are a couple of things that come to play here. One, first of all, to answer your question, we lose about 5%, okay, as it turns out, which is much better than 50% than I'd expected. But it's because we're working with people who know these animals really well, who have specific, very defined research questions. So while some deployments may go for a week, some go for 2 weeks, but in a way that's very predictable. The animals are expected to come back to the beach, or they're expected to be in this same habitat for that period of time. So and our collaborators know this well enough to be able to make these kinds of predictions. Increases the success rate considerably. And then, of course, on the technology side, constantly getting better at what we do. Back in the old days, every other camera was half flooded with water. I remember one I got back from a great white shark who did the deployment, and the Crittercam system comes off, the animal is floating at the surface. You see this because the camera's still running, and the shark swims, turns around in the distance, comes back and knocks the camera, bumps it a couple times, ultimately puts it in its mouth, and that's one of the possible. People ask me, do they ever eat them? Well, that's really the only example I have of even an effort. Spat it back out. But when I pulled that system out of the water, got it up on the boat, I always hold these things up like this to see if there's any water in them. And the whole lens is full of water. But fortunately, I held it like that, and the videotape itself was still dry, so I could get it out. But yeah, we've just gotten much better. So the technology's gotten better, and really our research collaborators are predominantly responsible for ensuring that we make sensible deployments. Yeah.
Yes. Is there any suspicion that the lack of mating data is to do with the changing appearance of the carrier animal?
Is there any suspicion that the lack of mating may be due to the change in appearance, the impact of the Crittercam system on the animal's back or wherever it happens to be? I would say that in the case of those leatherback turtles, it seems it's not unreasonable to think that may be the case. We really didn't expect that to see any mating behavior, so it was somewhat of a surprise. Now we did subsequently do 10, 15 more deployments, but again, just to reconfirm that what we were seeing was real. But there may have been an impact there. Now the weight of other evidence, the genetic evidence in the case of those leatherback turtles, suggests that the salmon, the sneaker salmon story. What happens is salmon, they look for robust mates, and it's a large male, large female that generally mate. But there are these small males, sneaker salmon, they're called the sneakers, that sort of hide and get into the nest and fertilize some of the eggs. And they're successful, and they are successful in reproduction. Now, the speculation is that because only 1% to 2% of the eggs that a given leatherback female lays are different, genetically different from the vast majority of the eggs, that somehow there are sort of sneaker males as well. And it may in fact be those males that we discovered just off the beach. It could be that the predominant mating happens out at sea, and that it's only these, and I'm making this up, you won't see this in the literature because it's speculation at this point. We need to do more work to confirm it, but it could be those sneaker male leatherback turtles that are following the females in off the pelagic zone onto the beach. And then, coitus interruptus, because you got the Crittercam system on their back, which is, so we don't know. It's a good question. Generally speaking, I don't think so. For most of the other species that we're working with, many species, we're looking at them during their foraging behavior, during other aspects of their behavior ecology, and not necessarily the mating season. Yeah.
Any thought to using your cameras on the underside of some of these marine mammals? I know it's going to be harder to attach.
Yeah.
But it might get you more into mating habits and other things that aren't visible when you're only looking at the back of the head of the...
Exactly. Is there any objectives to try to look at the underside of the animal, to deploy the Crittercam systems on the underside of the whales or many of the other species, in fact, as well? And it's a great question. It harkens back to the very beginning of my talk when I showed you the picture of the remora, and that the dream being that someday we'll be able to have this remora technology to work our way around the animals. The worst example, of course, is whales. I mean, gosh, you don't want to be in the back of the whale. The mouth is 15 feet down there, underneath the animal. We want to be down there seeing what's going on. The problem is, we're just not smart enough. We haven't figured out how to do it. We haven't figured out how to be able to reach a pole down into the water and effectively, in the water, achieve a suction onto the side of the animal. So it's a matter of creativity. We just haven't gotten there yet. I've only been at this 25 years. Give me another 25 years, we'll figure it out.